Okay, so now we're going to move on shortly to some other case studies that look at binding interactions. We've talked a lot about cooperativity. That was in case study number 1 and looking at hemoglobin and its types of interactions. But now we're going to move to how protein peptide interactions can be helpful and act in different ways. And in this case study, we're going to look at antibody and antigen interactions. Now, to understand these interactions, we're first going to review the idea of the immune system. Our immune system is made up of two immune systems, which is the cellular immune system and the humoral or fluid immune system. The cellular immune system is going to target our own cells that have potentially been infected. So, this is the immune system that's reserved for infection of the actual biological unit's own cells. This also works for viruses because, of course, it clears up virus particles that have been infecting cells and also bacteria that is infecting cells. So, in the case where you have some unit infecting the actual cells of that organism, the cellular immune system is still used. The key players, the key components of a organism that act in the cellular immune system are macrophages and killer T cells, which we'll abbreviate as this TC, and also some types of inflammatory T cells. So mostly T cells, as you can see, and macrophages. Now, the fluid or humoral immune system is going to target the actual pathogens. So anything that's outside of the cell but should not be within the organism. So again, we're not targeting our own cells in this system, but targeting the actual pathogens. It can help recognize foreign proteins, and generally it makes these soluble antibodies that then are going to help us continuously recognize pathogens as they enter. It helps organisms keep a memory of past infections. And this immune system is essentially what we're using when we develop vaccines, for example. The players in this type of immunity are our B lymphocytes and helper T cells. So you'll also see these B cells for the B lymphocytes. They'll be called B cells sometimes. So again, a review on the two types. In cellular immune system, we're still using antibodies, but the antibodies bind to fragments displayed on the invading cells. And then phagocytes, which are specialized cells, are brought to help essentially eat or engulf the invaders. Macrophages are a type of large phagocyte that helps ingest bacteria that are tagged by these antibodies. So again, the idea behind this is an antibody is going to tag the cell, the invader cell, or the cell within the organism that is been disrupted or has been compromised in some way. And then phagocytes or macrophages in general are sent to then engulf and destroy that cell. So we're using a type of antibody that recognizes some portion of the outside virus cell or whatever the foreign unit is. And then the other side of the antibody binds to a receptor on the actual phagocyte or the macrophage. So again, one side is binding to the foreign invader, the foreign cell, and the other side is binding to the macrophage to help the macrophage recognize where it needs to engulf.
Now, we also fight infections again with antibodies that are specifically made to bind antigens, which are just anything that is foreign. We describe an antigen as any substance that's foreign that stimulates the production of antibodies. So antigens, things that are foreign, are typically macromolecule in nature. They're typically large, and they're recognized as foreign by the immune system. They can be, for example, proteins that coat bacteria or viruses, or they could be something like the surface carbohydrates of cells or viruses. So they're typically a unit of the whole thing, some type of protein, some type of carbohydrate, some piece along the whole foreign invader that is going to be actually recognized. And of course, the antibodies themselves are proteins. Remember that antibodies are proteins. They're a type of protein, and they are made by B cells to specifically bind to antigens. So B cells will produce a protein that recognizes a piece of some foreign material, usually some protein on a bacteria or protein on a virus or a carbohydrate on a bacteria on a virus. And that antibody will then be able to bind. So again, the antibody binds a small region, just a small region, just maybe one protein or one carbohydrate or one unit of whatever the foreign invader is, whatever the antigen is. Now, one antigen, one foreign invader, could have several what we call epitopes, meaning that if this is your virus and it has different things on it, it has some sugars, it has some proteins, it has some lipids, maybe. A B cell could make multiple antibodies that recognize your virus. So a B cell might make an antibody that fits this particular protein, and it might also make an antibody that fits this sugar. And it might also make an antibody that fits this lipid. So there could be multiple antibodies or an antibody with multiple binding regions. So regions that can bind multiple things along the same foreign invader, the same antigen. So again, one foreign invader, one antigen could have several regions on it that our body will develop antibodies for. Antibodies have this multiple unit setup. So again, they are a protein that is made up of multiple peptide chains. So you can see the chains highlighted here. It actually has a total of these 1, 2, 3, 4, 5, 6 chains that are interacting in this quaternary structure. It's usually drawn as a Y. That's why we make antibodies drawn as a Y, is because of their shape. Now, these subunits are generally broken up into two areas called the light chains and the heavy chains. There are two heavy chains and two light chains, and they are generally composed of an area that is constant, so constant domain, and a variable domain. You can see the light chains have a constant area and a variable area, and the heavy chain has 3 constant areas and 1 variable area. So it's labeled here at the bottom. The constant areas are in C. Here's constant area, constant area. Okay, so we can see the light versus heavy chains of the, sorry, missed my constant areas here too, of the constant versus variable areas. And then the heavy and light are documented by H versus L. So for example, all of the light chain on this portion, this is light chain, and here we have light chain. And then the heavy portion is what's in blue, which is here. Now, the main takeaway from these antibodies is the areas that we call the variable domains and the areas that we call constant domains are areas that are either unchanging or essentially variable. Constant domains are unchanging. They're part of the protein in which its amino acid structure is not going to change, and thus the structure of the whole protein in that portion won't change. The variable domains are variable for exactly the reason that they need to change in order to fit whatever specific antigen, whatever specific foreign body they are trying to recognize. So the actual amino acids of that portion of the protein will change, will be able to change and be altered in a way that changes the secondary and tertiary structure of that portion of the protein in order to make it able to bind a new ligand and able to make it able to bind that antigen. Here's just a three-dimensional representation of a typical antibody. Immunoglobulin G is what we call this base structure of the antibody. And of course, each antibody, as I said, will have this portion here that is unique. Notice that antibodies actually have within their constant domain a carbohydrate that is bound permanently here. And this is, again, in this constant domain area at the bottom. Now, when an antigen binds to an antibody in that actual binding area, you can see that it induces a structural change to the antibody itself. So here is just a close up. This is just this region up here, which we call the FAB region. This is the area where you have antigen binding. That's where the AB comes from. So this is the antigen binding area. And at the bottom is the constant area, the area that's not changing. So you've closed up onto the antigen binding area here at the top of an antibody. And you can see when the particular antigen or ligand binds to that site, you actually have conformational changes to the antibody. And the conformational changes make the antibody bind more strongly to that antigen. So again, we talked about this in the first section, induced fit. So this is not lock and key type where the antigen and the antibody have a very, very perfect fit for each other. The antibody has a general fit for the antigen, but when it binds, it then changes structurally in a way that makes the fit even more strong. So it binds to have a stronger interaction with the antigen. Now, again, we spoke about this a little bit previously, but a reminder of how we use antibodies and their specificity in a way in biochemistry very often to run different assays. So it's used as an analytical reagent. It's used to help us recognize and analyze and get information from different samples. Because antibodies have this very high specificity, because again, when they bind, they have this induced fit. And so they bind very tightly and very specifically to that ligand or that antigen of interest. We can use them to help us recognize when we have a protein of interest or a molecule of interest in a sample. So again, generally, this is a type of study that is used to identify how much protein is in a sample. And again, this is a specific identification. So it's going to identify how much of one single type of protein is in our sample. And the way that we do this again is by first designing an antibody that has a binding site that is specific for whatever protein we want to look for. So we have some solution or some sample. Maybe this is in a tube and it has a mixture of things in it, but we want to know how much of our protein, this blue protein, is in that actual solution. So we'll first put an antibody that's designed to just bind our protein. And then after adding that, we will then incubate with a secondary antibody that is designed to recognize the first antibody that we put in. You can see it locks into the end of the first antibody that we added. This secondary antibody will have some type of probe on it. And this probe usually will form a color or it'll fluoresce. Usually, one of these two things, fluorescing or forming a color, will occur once you bind these antibodies together. And in this way, what we can do is just look for how much color is present or how much fluorescence is present, and that will tell us how much of the protein we're interested in is present in that sample. So again, this is a very, very, very important analytical reagent that we use, which is using antibodies to our advantage to help us understand more about samples. This is, again, the basis for a lot of tests that you receive. It's the basis for something like a pregnancy test. The pregnancy test sticks that you get actually have small amounts of antibodies on them, and they change color in relation to if they see a specific protein or not. Even the COVID-19 tests that are being developed for your blood are going to be having some antibody in them that change color if they recognize the protein related to COVID-19. Here's an example of one of those types of tests in which we use protein binding, antibody binding, to our advantage. It's called an ELISA. ELISAs use these plates in which you put the sample you have, a solution of protein. This could be blood, it could be whatever it is you're interested in looking at. And you incubate it with these antibodies. And you can see, for example, based on the color, how much in each of those wells, how much protein there is. You can see if there's protein and how much. So you can tell, for example, these ones I've circled that are changed to a yellow color, they have the protein in them and there's a lot of it. Whereas maybe a sample like this at the top, where it's kind of a lighter yellow, there's still protein in there, but there's maybe not as much. So we can use this color change and we can then actually analyze the color change with spectroscopy. Remember, spectroscopy helps us measure how much color there is. So we can use spectroscopy and then analyze a plate like this. And it will tell us, for example, your color change here is 3, your color change here is 1, your color change here is 0.5, and here it's 0. And this lets us quantify, gives us a number, an actual set of data to quantify how much protein is in a sample.